Picture your typical morning after, like a genuinely bad night's sleep.
Oh yeah, we've all been there.
Right. You look at the clock, you realize you've maybe scraped together 5 hours if you're lucky. Your head feels super heavy, your eyes are burning.
Just that classic brain fog.
Exactly. And you do what almost every single one of us does in that exact situation. You drag yourself to the kitchen and you pour a giant cup of coffee.
The universal fix.
Yeah, the totally socially acceptable fix for a sleep deficit.
Yeah.
But looking at the source material we have today, we really need to ask a much more physiological question.
Which is pretty terrifying, honestly.
It is. We're asking, what is that specific combination of chronic sleep restriction and daily caffeine actually doing to the physical structural tissue of your brain?
Because we tend to treat that morning routine like a really simple mathematical equation, minus sleep plus coffee equals a functioning human being.
Right, like it just cancels out.
Exactly. But we rarely stop to consider the actual biological transaction taking place there. I mean, we are introducing a really powerful psychoactive substance into a neurological system that is already severely compromised.
It's fighting to adapt. And the source we are unpacking for today's deep dive takes that biological transaction totally out of the realm of guesswork.
Yeah, it puts it right under a microscope. Well, an MRI machine, really.
Okay, let's unpack this. It is a highly rigorous, double-blind, randomized controlled study, and they used advanced PET-MRI brain scans to observe this exact scenario.
Which is huge.
Yeah. The mission of this deep dive is to look at this data and figure out if your daily caffeine habit actually protects your brain from the structural damage of sleep loss, or if it's, secretly undermining your brain's ability to survive the deficit.
And just a quick note for our listeners, we are focusing purely on gray matter today.
Right, the gray matter. So that's the actual processing centers, right?
Yeah, exactly. It's full of neuronal cell bodies, synapses, and dendrites. We aren't really looking at the white matter tracks today, which are the parts that just connect those centers.
It's just the processing hubs. And looking specifically at gray matter in a highly controlled environment like this is what separates this research from just simple observational studies.
Oh, absolutely. I mean, for years, we've had isolated data suggesting that both sleep loss and chronic caffeine consumption can independently alter gray matter volume.
Right, like separately.
Yeah, but combining them to observe the interaction under really strict laboratory conditions, that gives us a much clearer picture of what happens to, like, the typical corporate worker Monday through Friday.
Okay, so the setup of this experiment is basically a gauntlet designed to mimic that exact stressful work week. They took 36 healthy adults. Average age was around 29.
A pretty typical demographic for this kind of lifestyle.
Definitely. And they literally moved them into a laboratory for 9 days.
Wow.
Yeah, a 9-day commitment. They established a baseline first with 2 days of just luxurious 8 hours of sleep.
Sounds nice.
Right. But then the researchers imposed 5 straight days of chronic sleep restriction. They only allowed the subjects 5 hours in bed per night.
So they're building up a serious sleep debt.
A massive debt. And here's the split. 19 of these subjects were given 300 milligrams of caffeine daily.
Which is roughly 3 standard cups of coffee.
Yeah, spread out to mimic a normal working day. And the other 17 subjects, they received a perfectly matched decaf coffee.
The placebo group. And the double-blind nature of that split is vital, obviously, but so is the imaging technology they use.
The PET-MRI setup.
Right. They didn't just slide these folks into a standard MRI machine. They utilized a multimodal PET-MRI approach. And that allowed them to simultaneously measure cerebral blood flow right alongside the structural gray matter volume.
Okay, let me make sure I'm following the logic of the methodology here. Because caffeine is a known vasoconstrictor, right?
Yes, it physically narrows the blood vessels.
Right. So if they only used a standard structural MRI, a reduction in blood flow could simply look like the gray matter tissue had physically shrunk.
Exactly. It would look like shrinkage when in reality it was just temporarily starved of blood.
Ah, okay. So by running a PET scan simultaneously, they could mathematically sort of separate the blood flow variable from the actual physical tissue volume.
That is the exact reason for the dual imaging approach. I mean, it immediately rules out the most obvious counter-argument anyone would have.
It's bulletproof.
Yeah, they statistically controlled for the vascular changes. So they prove that any alterations they saw in the brain volume were genuine, actual structural modifications to the tissue itself.
That's wild. Okay, so with that baseline established, the scans of the decaf group provide the first major finding.
And this was super surprising.
It really is. So these are the 17 people surviving on 5 hours of sleep for 5 straight days, right?
Oh.
Relying purely on their own biology, no chemical assistance.
Just pure grit.
Right. And after 5 days of restriction, the scans showed their gray matter actually increased.
Yeah, it physically grew.
Which blew my mind. It specifically grew in regions like the thalamus and the prefrontal cortex, which, well, that handles our complex decision-making and focus, right?
Yep, the executive functions.
Wait, isn't sleep deprivation supposed to, like, destroy our brains?
Yeah.
Why is it growing? I mean, is this like lifting weights? Where the stress of staying awake forces the muscle of the brain to swell and adapt.
That's a really good analogy, actually. The biology behind that scaffolding relies heavily on what they call the synaptic homeostasis hypothesis.
Okay, break that down for me.
So to understand why the brain expands, we have to look at the daily life cycle of a synapse. During the hours you're awake, you are constantly processing stimuli, right? You're learning, you're reacting.
Taking in the world.
Exactly. And your brain manages all this by increasing synaptic strength. The connections between your neurons actually become thicker, they become more active, and eventually highly saturated.
Okay, and then sleep is the biological mechanism that prunes those connections back down, right?
You got it. It clears out the unnecessary noise and sort of resets the baseline for the next day.
So without that restorative pruning process, the architecture of the brain just fundamentally changes.
It has to. Now, in cases of total prolonged sleep deprivation, say, staying awake for 3 consecutive days, the brain eventually just hits pure cellular exhaustion.
It just crashes.
Yeah. The neurons cannot sustain the metabolic cost of that tension, and the tissue does begin to degrade. But remember, the subjects in the study were getting 5 hours.
Right. Partial restriction, not total deprivation.
Exactly. That partial sleep provided just enough restorative downtime to prevent total cellular collapse, but not nearly enough time to fully downregulate and prune the synapses.
So the synapses are trapped in this sort of biological traffic jam.
That's a great way to put it.
Because the restorative window is cut short, the brain just never fully clears the board. It leaves the tissue in a state of constant upregulated tension.
Yes, and the physical swelling of the gray matter we see on the MRI, it isn't necessarily healthy growth, it is the structural manifestation of prolonged stress.
Oh, wow.
Yeah, the brain is artificially expanding its processing capacity simply to handle the overwhelming deficit of being awake for 19 hours a day.
So it's basically surviving. The cellular demand must be massive.
Oh, it's huge. Maintaining that upregulated state requires constant energy expenditure just to keep the individual functioning at a really basic cognitive level. It's a highly taxing survival strategy.
Okay, so if the unmedicated brain relies on swelling its synapses to survive a 5-hour sleep schedule, introducing a chemical that blocks energy awareness completely changes that survival strategy.
Completely flips it on its head.
This brings us to the caffeine group, the 19 subjects receiving the 300 milligrams. In the exact same brain regions, the prefrontal cortex and the thalamus, their gray matter didn't swell.
No, it decreased. It actively shrank.
The structural response just completely flipped.
What's fascinating here is that the introduction of the stimulant initiated an entirely different cascade of neurological events.
Right. And reading through the biological mechanisms detailed in the study, I realized this really comes down to how caffeine interacts with adenosine.
Yes, adenosine is the key player here.
We talk a lot about adenosine as the brain's sleep pressure gauge, but fundamentally it's basically the cellular exhaust pipe.
Exactly. It is the byproduct of ATP, which is the primary energy carrier in our cells.
So when neurons fire, they burn ATP and leave behind adenosine.
Right. And when that adenosine binds to specific receptors, specifically the A1 receptors, it acts as a biological accountant.
Mm-hmm.
It signals to the brain that energy stores are critically low and it needs to shut down.
So it's the gauge. And if adenosine is the gauge that builds up while we are awake, and caffeine puts tape over that gauge so we don't feel tired, does caffeine also trigger an aggressive energy-saving mode in the background?
It absolutely does, because caffeine's molecular structure allows it to perfectly fit into those A1 receptors without actually activating them. It operates as a non-selective adenosine receptor antagonist, meaning it physically blocks the accountant from delivering the message.
So you don't feel the fatigue because the signal is just jammed.
Right. But the biological reality of the energy deficit remains entirely unchanged. Your neurons are still burning ATP, they are still exhausting their fuel.
But the brain's internal monitoring system is totally blind to the crisis.
Exactly. And because the brain is forced to stay awake while running on an empty tank, it initiates a severe background energy-saving mode to protect itself from cellular death.
Oh, wow. So it just shuts non-essential stuff down. And the study points out that the primary way it conserves this energy is by shutting down synaptic plasticity, specifically halting a process called long-term potentiation.
Yes. Long-term potentiation, or LTP. It's the fundamental mechanism the brain uses to strengthen synaptic connections.
Which is how we learn, right?
Yes, it is the physical basis of learning, memory consolidation, and basically adapting to your environment. But the mechanics of LTP are incredibly resource-intensive.
Like a construction site.
Yeah. To strengthen a connection, a neuron has to synthesize new structural proteins, transport them down the axon, and physically build new dendritic spines. It is a really expensive construction project.
And when caffeine blinds those A1 receptors during chronic sleep restriction, the brain recognizes it cannot sustain both consciousness and construction.
So it halts the protein synthesis.
It just stops building the dendritic spines.
Yep. The swelling and adaptation we saw in the decaf group, that is completely shut down.
And that active suppression of the brain's structural remodeling is what actually manifests physically on the PET-MRI scan as a decrease in gray matter volume.
Exactly. The caffeine is basically forcing a biological triage. It maintains baseline alertness, but at the direct expense of structural adaptation and synaptic strength.
So you are effectively borrowing structural integrity from your brain just to pay for a few hours of artificial wakefulness.
That's a perfect way to look at it.
But looking at the data, the structural shrinkage isn't uniform across all 36 subjects, is it?
No, not at all.
The study dives into this really fascinating layer of individual variation. It shows that genetics play a massive role in how much gray matter you actually lose in this scenario.
Yes, and to measure this, the researchers utilized a highly specific radioactive tracer during the PET scans.
Right, a compound called 18F-CPFPX. Try saying that 5 times fast.
I know, right? But the application of that specific tracer really elevated the entire experiment. Instead of just observing the macroscopic changes in tissue volume, the tracer allowed the research team to map the microscopic chemical architecture of each participant's brain.
Because the tracer binds exclusively to the A1 adenosine receptors we were just discussing.
Exactly. By using the tracer during the baseline rest days, they essentially counted how many A1 receptors each individual naturally possessed before any sleep deprivation or caffeine was ever introduced.
Oh, that's clever. So some people naturally have a dense population of these receptors, while others just have significantly fewer.
Yeah, it's just a genetic lottery.
So are these A1 receptors basically acting like the brain's shock absorbers?
I love that analogy.
Yeah. If you're naturally born with fewer shock absorbers, does the caffeine bump do more structural damage to your gray matter?
It does. If the A1 receptors are the biological brakes that sense exhaustion and manage energy, the people with a high baseline count basically have a heavy-duty suspension system. They have more shock absorbers to handle the impact of sleep loss.
Okay, so to stretch that analogy, taking 300 milligrams of caffeine during a state of sleep deprivation is like driving that vehicle over a massive pothole.
Yeah. And if you naturally have an abundance of shock absorbers, the caffeine will block a significant portion of them, sure, but you will still have enough unblocked receptors left over to receive the adenosine signals.
So your brain retains at least a partial awareness of the energy deficit.
Right, and it can manage the stress without completely shutting down structural maintenance.
But if you were born with a lower baseline of A1 receptors, you are hitting that same pothole with a really fragile suspension.
Exactly. The standard dose of caffeine effectively blocks almost all of your available receptors.
Wow.
So your brain's energy regulation system is entirely blinded, and that triggers a much more aggressive and severe energy-saving shutdown.
Which results in a significantly larger drop in gray matter volume.
Yes. The researchers documented this as a concentration-dependent switch located primarily in the striatum.
The concentration-dependent switch.
Yeah. The natural density of your A1 receptors serves as a biological buffer. It dictates your personal resilience to the combination of chemical masking and sleep restriction.
That is wild to think about. Two people can work the exact same corporate schedule, drink the exact same amount of coffee, and experience vastly different levels of structural degradation in their prefrontal cortex.
Based entirely on a baseline receptor count they have absolutely zero control over.
Wow. Well, I'm sure anyone listening right now who relies on a morning coffee after a terrible night's sleep is probably having a mild panic attack.
Yeah, wondering if they've permanently shrunk their brain.
Exactly. Have they permanently damaged their cognitive architecture? But thankfully, the study directly addressed the reversibility of this process.
It did, yeah.
After the 5 days of restriction, the subjects were given a single recovery day. This meant an 8-hour sleep opportunity, which, for the caffeine group, also equated to roughly 35 hours of complete caffeine withdrawal before the final MRI scan.
And the data from that final scan provides a really remarkable look at the elasticity of the human brain.
Right.
For almost the entirety of the cortex, the plasticity was fully reversible. The upregulated swelling observed in the decaf group, as well as the suppressed shrunken gray matter in the caffeine group,
It all bounced back.
it returned precisely to their original baseline volumes after just one night of restorative sleep.
So the tissue bounced back. The brain recognized the energy crisis had passed, lifted the restriction on long-term potentiation, and just resumed normal synaptic maintenance.
Yep, it got right back to work.
The study highlights one critical, stubborn exception where the gray matter did not recover.
And this is a big one.
The thalamus. The thalamus of the caffeine group remained significantly reduced even after the recovery night. And I've got to push back on this a little bit. Is one 8-hour night genuinely enough time to recover from 5 straight days of restricted sleep? I mean, why is the thalamus being so stubborn compared to the rest of the brain?
That's a totally fair question. If we connect this to the bigger picture, the thalamus is indeed operating under a unique set of metabolic constraints. It's not just another cortical region, it is the central relay station of the brain.
Okay, what does that mean exactly?
Well, almost every piece of sensory information aside from smell actually must pass through the thalamus before it reaches the cortex for processing. But even more importantly for our topic, it is also the primary engine regulating the sleep-wake cycle itself.
So because it is the grand switchboard operator, it never truly powers down.
Exactly. The rest of the cortex can enter deep restorative states and rapidly flush out all that accumulated adenosine. But the thalamus is perpetually engaged in monitoring the environment and managing the actual architecture of your sleep stages.
So that relentless functional demand means the thalamus has a significantly slower metabolic timeline for recovery.
Yes. The study noted that adenosine signaling in the thalamus continues to drop for hours, even well into a recovery sleep phase. It just cannot simply flush the system and reset in a single night.
Wow.
So when you combine the physical stress of chronic sleep restriction with a chemical antagonist that specifically targets the sleep regulation receptors.
You are placing the highest possible metabolic burden on the exact organ trying to save you.
Right. The central engine gets overloaded. The rest of the brain might bounce back quickly, but the thalamus requires a much longer runway to repair the structural suppression caused by the caffeine. The damage really lingers at the very core of your sleep regulation system.
Which creates a super dangerous physiological loop, right? If the thalamus is structurally compromised and struggling to recover, your subsequent nights of sleep may be less efficient.
Which leads you to reach for even more caffeine the following week.
Continuously compounding the stress on that specific region. That is brutal.
It's a tough cycle to break.
Taking all of this deeply sophisticated imaging data and applying it to your daily life really changes the way you should look at your morning routine.
It definitely should.
I mean, the next time you manage only 5 hours of sleep and you brew a strong cup of coffee, you have to recognize the biological trade-off you are making.
Yeah, the caffeine is absolutely going to mask your fatigue.
Right, it'll allow you to function through the morning meeting. But beneath that artificial alertness, it is actively suppressing your brain's physical attempt to adapt to the deficit.
It's forcing your neurons to abandon the structural maintenance of your synapses just to keep the lights on.
And it is placing a lingering, disproportionate strain on your thalamus.
The chemical intervention has a tangible structural cost. You are basically trading long-term cognitive adaptation for short-term wakefulness.
Which leaves us with a highly provocative thought to consider, especially for anyone navigating a high-pressure academic or corporate environment. We are all familiar with the classic all-nighter.
Oh, absolutely. Red Bulls, coffee, staring at a screen.
Exactly. You're a student cramming for a final exam or a professional staying up until 3 in the morning to memorize a massive presentation. You continuously drink coffee to fight off the sleep pressure,
Yeah.
specifically so you can learn and retain the material right in front of you.
But the mechanism of long-term potentiation we just discussed makes that strategy incredibly counterproductive.
Exactly. If the study proves that caffeine, combined with sleep restriction, actively forces the brain to halt long-term potentiation, which is the exact physical process of building new dendritic spines and strengthening synaptic connections to form memories, what are you actually accomplishing at 3 in the morning?
You're shooting yourself in the foot.
Could the very chemical you are consuming to stay awake be physically paralyzing your brain's ability to build the neural infrastructure required to remember the information you are studying?
That's a really good question.
You might be forcing your eyes to stay open, only to logically lock the doors to your own memory centers. So that is something for you to mull over the next time you find yourself staring at the bottom of a coffee mug after a terrible night's sleep.